artificial radioisotopes which is complementary to VNTR, and then excess radioactive probe is washed, means removal of excess. Fine. So now I have this plate. That plate consists of VNTR like this, and complementary to the VNTR is the radioactive probe like this. Fine. So VNTR, artificial radioactive isotope is complementary to the VNTR, right? And when we are going to pass this plate with the VNTR and the artificial radioactive isotope through the X-ray plate like this, so this artificial radioactive probe, which is complementary to VNTR, this will create radioactivity just because of the isotope, and this radioactive radiation will create impression on these X-ray plates, this particular X-ray plate, and this radioactive impression will show DNA pattern. See here. Developing X-ray film to detect the radioactive probe like this. So on this X-ray film, we are going to see this pattern of DNA. And this DNA pattern is what? It's a DNA fingerprint. Yes, so again, repeating once again. See, isolation of DNA, then digestion of DNA through restriction endonuclease, then separation of DNA through the electrophoresis, then transferring of DNA to the nitrocellulose paper, the southern blotting, then hybridization of this VNTR to the radioactive probe, fine, then removal of the excess material, then developing X-ray film, right, and then just show the DNA pattern. So all these steps are mentioned here like this. We can see here. Isolation of DNA, digestion of DNA by restriction endonuclease, separation of DNA fragment, transferring southern blotting, fifth is the hybridization of VNTR, and the sixth is the detection of hybridized DNA fragment by autoradiography. Autoradiography means what? It's an X-ray technique. X-ray technique. So suppose this is my fingerprint. Suppose this fingerprint, what we have done so far is my fingerprint, right? And suppose if in my case there's a parental dispute. Means my either the parent is not willing to accept me, and they are not claiming that, they are just claiming that I am not there. They are not accepting me as a son. So in that case, I could claim as a, means I will go to the court and ask for a DNA fingerprinting, because DNA fingerprinting is the only biological pattern. Because if I am the descendant of my biological parents, so definitely there must be the continuation of even the similar pattern of the VNTR between me and my parents. So in that case, see what is going to be done. See, listen. This is my DNA pattern. This is my DNA pattern like this. This is my fingerprint. Yes, this is the DNA of individual A. Suppose it is my mother. DNA of individual B. Suppose my father. So here they are going to make a print of my father and mother. So, and then they are going to match. Like see here, the matching of this pattern, matching of the band, matching of the band, matching of the band, matching of the band. Clear? So if I am going to match my DNA band, at least one band with either of the parents, so that will show the biological connection between C means me, A means my mother, and the B means my father. So in that case, they are not able to justify their claim like I am not the product of these two individuals, because somewhere I am showing right similar DNA band pattern in VNTR to my parents, and that is considered as a biological proof. Fine. So that is the reason behind DNA fingerprinting is the only proof that we could never challenge, and this is considered as a biological proof. That is considered as a biological proof. Right? The similar case is also reported in the case of the criminal cases, like suppose in the case of the in the case of in rape cases. Fine. So from the female body, we are just trying to collect the traces of the semen. Semen is what? Semen is the good source of the sperm cells, right? So again, we are going to separate the different type of sperms from the female body, and then we are going to develop profile, means the DNA profile, and that DNA profile will act like a reference DNA sample, and then we are going to match this reference DNA sample with the accused DNA sample, and if this is going to match with the accused DNA sample, so that will show the biological connection, that will show the involvement in this crime, because the biological reference is already being collected from the body of the female. So that is the biological connection between that accused and this victim. Clear? And that is the reason behind this. Once we are going to claim as a, once we are going to punish as a capital punishment, so before the capital punishment there must be the clear correlation between the accused and the victim. Otherwise, right, the physical evidence could be forced, the chemical evidence could be forced, but biological evidence is usually, it is not being forced unless the procedural, there is a procedural fault, right? Otherwise, if there is no procedural fault and everything is all right, in that case we could not challenge this DNA fingerprinting as a procedural fault. So I hope you are okay with the concept. And let me just hold. I am just trying to show you the animated version of this. Probably I have the animation of the DNA fingerprinting. So I am just trying to run. Here, you know.
Biological tissue. At Guy's Hospital in London, this research worker is using an automatic DNA extractor to produce a high purity DNA fraction from a...
Dear students, I am just trying to play this animation. You please let me know whether you are going to get this animation or not. Are you able to see this animation, what I am playing? If yes, please let me know.
Blood sample. A restriction enzyme is added to the DNA. This enzyme moves along the DNA strand, cutting it at the so-called restriction sites. The restriction sites available to enzymes are located throughout the chromosome, except in sections consisting of copies of the core sequence.
Visible.
This process produces many pieces of DNA, which
Hold.
consist of repeats of the core sequence.
Let me show from
The
the
mixture
beginning.
of these DNA fragments sample of biological. A genetic fingerprint begins with a sample of biological tissue. At Guy's Hospital in London, this research worker is using an automatic.
Let me show. Okay, right. So let me make understand. Let me make understand. This is the entire process of the DNA fingerprinting that I am trying to play here. This is the DNA sample. This is the DNA sample. See here, it's a DNA sample. It's a DNA sample. Now this DNA sample has a restriction. See, this Caesar is like restriction endonuclease, and this restriction endonuclease is cutting the similar gene sequences, right? So it's a removal of the similar gene sequences. Fine. So restriction endonuclease enzyme is going to remove the similar gene sequences, and now what we have? Repetitive sequences, CORE. See, these are the different segments. These are see, probably, see, maybe there could be see, I am just turning an animation from my side, so there could be the video stuck. There could be the case of video stuck. Fine, because this animation is running. Animation is going on. It's not a static. So in that case, if you're going to feel like the video is stuck, so just bear with me at least for the next 2 minutes, because I'm just running animation just to make you understand this process of the DNA fingerprinting, right? So these are the different DNA segments. These are the different DNA segments, and these are the different DNA segments with the variable length. And these variable length are showing as a similar gene sequences, similar base pairs like the CORE, that make a sense of repetitive sequences or satellite DNA. And these are VNTR, variable number of tandem repeats, because the similar gene sequences are being repeated every time. That make a sense of variable means 1, 2, 3, 4. Number of tandem repeats means the similar gene sequences are being repeated every time. That is what it's a VNTR. Yes, fine. Now, from the DNA solution, now the DNA is being poured on the gel electrophoresis. This is what it's a gel electrophoresis, and here it's a DNA solution. It's a DNA solution, right? So once we are going to pass current, like this, we could see here, see, it's a movement of DNA. We could see here the movement of DNA. See, the red strands are nothing but it's a VNTR sequences. It's a VNTR sequence. It shows the separation of VNTR. Now it is being washed properly, and then transferring this VNTR sequence from the gel to nitrocellulose paper. This technique is the southern blotting technique. Then washing it, and then treating with the molecular probe, that is artificial DNA, and then it is passed through autoradiography. This is what? It's a DNA pattern. It's a DNA bank. Right? It's a DNA pattern or it's a DNA bank. Fine? It's a DNA pattern or it's a DNA bank. And once this pattern is established, suppose it is my pattern, the similar pattern they are going to they are going to develop for my parents, and then they are going to match like this what we have mentioned here. What we have mentioned here. Right? This is what it's a DNA fingerprinting. Nikhil, so many times I have repeated this VNTR, variable number of tandem repeats, means that DNA sequence that consists of repeated bases, that consists of the repeated base sequences, fine, that is the V, variable number of tandem repeats, means the similar gene sequences are being repeated throughout is a VNTR, or the satellite DNA, or the repeated DNA sequences. Fine, okay. So the DNA pattern is always unique. DNA pattern is always unique for everyone. That's why nowadays we are talking about, and we have so many applications of the DNA fingerprinting, right? Suppose when we talk about application of this DNA fingerprinting. So this technique is used for the DNA analysis in the forensic test, paternity test, and even it is also used in evolution. So here the main analysis is what DNA analysis in the forensic test and in paternity test. Fine, and even the DNA technology bill, the DNA technology regulation use and application regulation bill. This bill was tabled in Parliament in 2019. And here, the intention is what? To create a regulatory framework for obtaining, storing and the testing of DNA sample of the human being, mainly for the purpose of criminal investigation with the objectivity and establishing the identity of a person. The proposed law seeks to bring in a supervisory structure so that the DNA technology is not being misused. Means the point is that suppose if we talk about the physical evidence could be forced, any kind of evidence could be forced, but if the DNA sample is if the DNA sample is stored and if the DNA fingerprinting DNA sample is stored, and if the DNA fingerprinting is already been stored in the system, so that can never be forced. Like example, suppose if I made a crime and I simply replaced my face, I have grafted my face through the plastic surgery. So in that case, I am going to have a new face. But if my DNA is stored, so if someone is going to match the DNA of my previous face and the DNA of my new face, in that case, they are going to see the genetic DNA similarity between these two faces. So ultimately, they are going to help me for the commitment of this crime, right? So that is the logic behind why this test is compulsion for especially for the criminal investigation, because in the criminal investigation, if the DNA sequence or the DNA sample of criminal is stored, it is already been matched, it is already been customized in the system. So in that case, they are not going to make any kind of the fraud, clear, and they are not going to make any kind of fraud is the ultimate purpose of this DNA technology and the Regulation Bill 2020. And there are different provisions. There are different provisions in this bill. And just look at the provisions. This bill regulates the use of DNA technology for establishing the identity of a person in respect to the matter listed in schedule, criminal matters and the civil matters. Fine. This bill establishes a national and the regional DNA data bank. Every data bank will maintain the following indices like crime scene index, suspect or under trial index, offender index, missing person index, and the unknown deceased person index. Means 5 index they are supposed to maintain. This bill establishes a DNA regulatory board. Every DNA laboratory that analyzes a DNA sample to establish identity of an individual has to be accredited by the board. Means, this is not supposed to be done by the independent DNA lab. Rather, the entire storage, processing, and the entire storage and the entire process of DNA fingerprinting and even the data storage is done only in those DNA lab which is accredited by board, means which is authorized by board, because the data leakage is the biggest concern. Data security is the biggest concern. So private agencies are not supposed to perform this unless they are specified by government or DNA regulatory board. They are competent for this kind of a job, because whosoever is going to perform this action, right, that agency must be responsible for, right, that agency must be responsible for all kind of the odds. If someone is going to, if there is a mishappening of the data, there's a leakage of the data, there is a stolen of the data, there's a misuse of the data. So in that case, that authority, means that lab will be responsible for this kind of. So in that case, that is the reason behind only the deserving and accredited lab is going to perform these kind of actions what we are talking about. Yes, the written consent is required from the individual to collect DNA sample from them. Means they are not going to ask you for verbally for the DNA sample, rather they need your written consent. Fine. The consent is not required for the offense with the punishment of more than 7 years of the imprisonment or death. Means the written consent is required by everyone except those individuals. They are penalized with more than 7 years of the punishment. But they are penalized with more than 7 years of the imprisonment or capital punishment. So for individuals, for anyone whosoever is penalized with more than 7 years of the imprisonment, consent is not required, or whosoever is penalized with the capital punishment, consent is not required. Otherwise, consent is required by individuals less than 7 years of the imprisonment or even the normal individuals. Right? So consent is required like that. So these are the different provisions of DNA Regulation Bill 2019. Fine. So there was a question, that question was asked by I'm not able to recall who asked this question. Someone asked this question, why erythrocytes are not used in DNA fingerprinting. Yes, this question was asked by someone else. I am not able to see who asked this question. Anyway, I am just answering this question. See, when we talk about the erythrocyte RBC, the mature RBC does not have nucleus. Mature RBC does not have nucleus. Means in mature RBC, there is no genetic material. Fine. That's why when we talk about the blood cell, it means we are simply talking about the involvement of the WBC in the process of DNA fingerprinting. Only WBC means the white blood corpuscles are involved in the process of DNA fingerprinting. So whenever we talk about the blood cells are being used in DNA fingerprinting, it means we are simply talking about the involvement of the WBC. not RBC, because mature RBC does not have nucleus, and there is no nucleus means there is no genetic material. Yes, Asif, I hope you understand. You got your answer. Jasna Isaac, sir, if the person have two types of DNA when bone marrow is replaced, fine. Jasna, see what happened. I told you bone marrow transplantation is a kind of somatic therapy. Fine, somatic therapy, right? So say, suppose if my bone marrow is not working, my bone marrow is not working, my original bone marrow is not working. In that case, you are not going to change my bone marrow. You are not going to change my bone marrow, right? You are not going to change my bone marrow. My original bone marrow has already been there. Original bone marrow is already been there. Now you are going to substitute my original bone marrow with the new bone marrow. So original bone marrow has already been there. That is quite inactive, and the new bone marrow is already been transfused to the body, and that bone marrow is going to develop, is going to form blood. So it is not like that. 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 First of all, we are removing original bone marrow and then we are substituting new bone marrow. It's a simple substitution process. Means simply transfusion of the new bone marrow to the person that already has bone marrow, existing bone marrow is not functional. So that is the standby, right? And that is not going to disturb your original gene sequences. That is the logic behind why we have mentioned in the gene therapy, somatic gene therapy. It is non-inheritable, it is temporary because the effect is restricted to that particular generation and it is not going to alter the original gene sequences of an individual, right? So it's a standby, and if it is not going to alter the original gene sequences of an individual, it means what happened? It is non-heritable in nature. So fine, that is non-heritable in nature. So somatic gene therapy or the stem cell technology is not being carried forward from one generation to other generation because it is not going to alter the original gene sequences of individual. Rather, it is a standby arrangement, and the standby arrangement is not affecting the original gene sequences. I hope you understand. Jasna Isa. Right. There's no RNA. See, there's no RNA fingerprinting. It's only DNA fingerprinting. Fine. Yes? Done? So this is all about the genomics, what we have done so far. So allow me to proceed. Allow me to proceed. Can we go ahead? If yes, allow me. Getting confirmation from the Nikhil Singh on behalf of everyone. No need to elaborate like this. See here. See, this already we have discussed. So ultimately, if you just look at that already, the application of this application of genome sequencing, है ना? See, no need to elaborate like this. See here, the population, structure and evolutionary history. Fine. Again, specific sequences for strain detection, है ना? Like COVID example. Yes, insight for the human growth. Yes, diagnosis, like see the real time PCR, है ना? DNA microarray. Then again protein expression, है ना? Which is used as a drug like insulin. Insight into the virulence means the point. See, already we have discussed. We are simply summarizing here. The application of genome sequencing means application of the genomics. So application of the genomics is for the improvement of the crop. Application of the genomics is for the the diagnostics. Application of the genomics is what the population structure and the evolutionary history. The application of the genomics is what different varieties means we are talking about the mutant varieties of the COVID. It is only after genome analysis, right? Expression of the genomics is what? Just to use this concept for the medication, like the protein medication, like the insulin therapy. So these are the different applications. These are different applications of the genomics that we are talking in a very simple ways. Fine, okay. So this is what it's application of biotechnology in the health sector and the application of the biotechnology in agriculture sector that we have discussed as a part of genetically modified organism or transgenics or Bt cotton, the Bt brinjal. Fine. Again, when we talk about application of this biotechnology in industry. Application of biotechnology in industry is the biotechnology application of biotechnology for the industrial purpose. And here the application is what? By using renewable raw material to produce a variety of the chemical and the fuels. Like here, the best example is biofuels. And the intention is what? Actively advancing towards lowering greenhouse gases emission and moving away from the petrochemical based economy. So here the ultimate purpose means ultimate point of the discussion would be the biofuel. And the intention of the biofuel is what? Moving away from the petroleum based economy. Moving away from petroleum based economy. Yeah, the point is that, listen, Asif Ali, yeah, you 100% are correct. I am talking about, in this context, what we are talking about, in the way we are talking about, in that context, there is no RNA fingerprinting. Otherwise, RNA fingerprinting is existing for the other aspect, but in this process, means in this casual aspect, what we are talking about, this concept is, means the concept of the RNA fingerprinting is not applicable. is not applicable in the case of what we are talking about. Otherwise, RNA fingerprinting is a technical aspect. It is a technical aspect, right? And this technical aspect is deserving. This RNA fingerprinting is technical aspect, and this technical aspect is also, required for the genome analysis of the pathogens and all and all. But now it is not required. That's why we are simply ignoring. And even for the crime detection, the RNA fingerprinting is not applicable. That's why I told you there is no RNA fingerprinting, especially in this context, right? So here, this biofuel, see, this biofuel, any hydrocarbon fuel which is produced from an organic matter, living or once living, is biofuel. This could be solid, liquid or gaseous. Solid biofuel, wood, dried plant material, manure. The slide is already with you. This slide is already with you. Liquid is the bioethanol and the biodiesel that we are going to elaborate. And the gaseous is the biogas. And the main reason for the shifting of the biofuel are the rising price of the oil, emission of the greenhouse gases from the fossil fuel, and the interest for obtaining fuel from agricultural crop from the benefit of the farmers. This is the intention ultimate. So here we need to discuss about the different types of biofuel, and especially different types of biofuel. So listen. Especially when we talk about the biofuel, we are talking about biodiesel and bioethanol. Biodiesel is mixed in diesel, and bioethanol is mixed in petrol. Bioethanol is the product of fermentation. And the bioethanol is technically, this bioethanol is technically C2H5OH, which is mixed with the petrol, while biodiesel is technically alkyl esters, and it's a result of trans esterification. It's a result of trans esterification. So biodiesel is technically alkyl esters and it's a result of trans esterification, and the biodiesel is mixed in diesel, fossil diesel, while the bioethanol is technically, it's a ethyl alcohol that is the result of fermentation, and this is mixed in petrol. It is mixed in petrol. Fine. So we have so many generations of biofuel as a whole. Look here. Like this first generation of the biofuel. And the source is what? Here the biofuels are, the source of the biofuels are the sugar, starch, vegetable oils, animal fat, conventional technology. Fine. And this includes bioalcohol, biodiesel, or even the vegetable oil. But it is not been promoted. Just because it will ensure that it will, if you are going to promote, so it will cause imbalance in food economy leading to increased food prices and the hunger. So it is being discouraged. It is not being promoted, means it is being discouraged. So what is being promoted? It's a second generation. So second generation of the biofuel is what? That is the produces from the non-food crop, which is not edible, like the waste, stem, husk, wood chips, fruit skin and the peeling, right? And even the food crop which is unfit for consumption, food crop which is unfit for consumption. These fuels do not affect fuel economy and their production is quite complicated. Their production is quite complicated, and see here. Anyway, I'm just going to elaborate. Here, I'm just mentioning here. Food crop unfit for consumption. Food crop unfit for consumption. Second generation, right? This third generation, algae. This is the research aspect, and here this is what? It's a butanol. It's a research is going on for this. Fine, research is going on for this. And the fourth generation is what? Here it's a sequestered, means this fourth generation is what? It's a carbon neutral. That is the product of climate engineering. This is the product of climate engineering. So as a whole, there are 4 generations of the biofuel what we have discussed, right? These are the 4 generations of the biofuel, and we are promoting this second gen. Nowadays we are promoting the second generation of the biofuel. But listen, if you just look at the Government of India biofuel policy, so as per the Government of India biofuel policy, there are 3 generations of the Government of India has categorized biofuel in 3 categories. Fine. So if you just look at the 2018 policy of Government of India, this is the National Biofuel Policy 2018. So this policy says that here, this policy says that there are 3 generations of the biofuel. One is the basic biofuel, that is the first generation. Second is the advanced biofuel, that is the second generation. And third is third generation of the biofuel. Right? Fine. And this policy, it expands the scope of the raw material for the ethanol production by allowing use of sugarcane juice, sugar containing materials like the sugar beet, sweet sorghum, starch containing material like corn, cassava, damaged food grains like wheat, rice, broken rice, rotten potato, unfit for the human consumption for the ethanol production. Means this is promoting second generation, right? Second generation. And as per this policy, the objective is reaching 20% of the ethanol blending and the 5% of the biodiesel blending by 2030. This is the target of this policy. Means we are supposed to ensure the 20% of the ethanol blending by 2030. Means in petrol, the 80% part would be the fossilized petrol, and the 20% part would be ethanol. And in the diesel, 95% would be the fossilized diesel, and the 5% would be the biodiesel. That is the target we are trying to, we have fixed to achieve by 2030 to 2030. And few means 2019-20 data is saying that now, as of now, we have achieved the 8% till date. We missed for the last 2 years. We are lagging behind for the data, and just because of the corona. Otherwise, 2 years back, the data was we achieved 8% of the ethanol blending in petrol. Means we have achieved 8%. Means still we need to ensure the 12% more of the mixing in petrol. Fine. So by 2030, we need to ensure that 20% of the ethanol blending in this. And see, in 2020, this question was asked. This question was asked in 2020. See here. As per the National Biofuel Policy, which of the following can be used as a raw material for the production of the biofuel? Yes, and it is carbohydrate material. Clear? So, cassava is a carbohydrate material. Damaged wheat grain is the carbohydrate material. Clear? Groundnut and the Hass gram, these two are fat and protein that we need to discard. Rotten potato is carbohydrate, starch, and the sugar beet is carbohydrate. So this is what the first option is correct option. Right? Okay. Right. And one more aspect is here in the same context, there is a concept of the oil japping. It is the bioremediation process which involves oil japping is what? That is the removal of oil from sea or marine surface. So it was implied to clean the Mumbai shoreline affected by oil spills that occurred in 2010. Yes, and it was the conduct of TERI, Tata Energy Research Institute. Fine. So how does oil japper works? This oil japper consists of 5 different bacterial strains. Clear? It's a mixture of 5 bacteria, and it feeds on the hydrocarbon compound present in the crude oil and the hazardous hydrocarbon waste generated by the oil refineries, and it converts them into harmless CO2 and the water. Means breakdown of the hydrocarbon of the oil, and then turn it into carbon dioxide and water. So by doing this process, we are going to clean oil on the marine surface, and that leads to cleaning of the oil from the marine surface is the concept of the bioremediation. And this bioremediation is done by the oil japping bacteria, and these bacteria are nothing but it's a group of 5 bacteria which is required to ensure this oxidation process is required for the cleaning of oil from the marine surface. So this technique was used successfully, and 1,30,000 means 1,30,000 tons of the oil/contaminated soil was treated as per the leading news sources, and the oil japper is neatly packed into sterile polythene bags and the sealed aseptically for the safe transportation, and the shelf life of this product is 3 months at the ambient temperature, means even it is available. It is available for the commercial exploitation that we can use, and here the shelf life of this entire package is a 3 month, and that is used everywhere. Means we can use it anywhere where there is a water clogging, especially because of the oil pollution. Fine. So listen, what we have done, let me conclude what we have done. Let me conclude here. This is the industrial use of biotechnology. Industrial use of biotechnology, and the ultimate purpose of this industrial use of biotechnology is the biofuel. Fuel which is biological in nature, and we have 4 generations, means the biofuels are categorized into the 4 generations. These are the different generations of the biofuel. First generation, the source is the food grains, means edible food product, which has not been permitted. The second generation is the municipal waste and the food grains which is unfit for the consumption, which is being promoted. Fine. This third generation is the biofuel, and the source of biofuel is the algae, and the fourth generation is the carbon neutral. Only this much of information is required for you, and then again at the same time we need to consider the national biofuel policy of the Government of India, which has classified this biofuel into 3 generations. The first generation is the basic biofuel. That again the basic biofuel is the first generation. That is the food grain which has not been permitted, which has not been promoted just because of the apprehension about the food scarcity. Second generation is the advanced biofuel, is the bioethanol and the biodiesel, right? And the source is what? It's a non-edible food products or food grains which is unfit for the consumption. And the third generation is what? Third generation of the biofuel as an algal biofuel. So in India, we are still promoting second generation of the biofuel, and just to promote second generation of the biofuel, right? The policy is already been modified, and in this policy, in this policy, there's an inclusion of few more sources as a source of the raw material, and these are the sources of the raw materials like the sugar beet, sweet sorghum, starch containing material, and all for the second generation. Why? Because we need to ensure the mixing of bioethanol 20% by 2030 in the petrol, and 5% of the biodiesel by 2030 in fossilized diesel. And here again at the same time we also discuss about, for the cleaning of oil from the marine surfaces, we are using a group of 5 bacterial strains, and these 5 bacterial strains are called as the oil zappers, and these oil zappers are responsible for the breakdown of the oil on marine surfaces, right? And this converts, it ensures the breakdown of oil hydrocarbon into the carbon dioxide and water, and as a result of this it is going to clean oil from the marine surface. And first of all, it was experienced in 2010 in the Mumbai shoreline, and that oil zappers was made by TERI. Only this much of information we are supposed to discuss in the environmental and the industrial application of the biotechnology. Apart from this, nothing is required. Yes. Right? So I hope you are okay with the discussion? oil japper is oil eating bacteria, right? So the group of bacteria is responsible for the eating of oil from the marine surfaces called as the oil japping bacteria, and this technique is the oil japping bacteria, and this is what this is called the oil japper. So there are 5 groups of bacteria which ensures breakdown of hydrocarbon of oil into carbon dioxide and water, and as a result of this breakdown, oil is going to be clear from the marine surface, and that is the way to handle, that is the way to clean all surface through the involvement of the biological process. So that's why this is called as this is called as bioremediation, means the cleaning of oil surface through the biological process. Okay. So I hope this is the end of today's lecture. Please